So with this video, we'll wrap up looking at the structures that are involved in the construction of eukaryotic cells, and then talk a little bit about how eukaryotic cells evolved from prokaryotic ancestors. So external structures that are found on eukaryotic cells include two structures that are responsible for locomotion. There are flagella and cilia. Flagella are in eukaryotic cells are much more complex and they're larger than prokaryotic flagella. And unlike prokaryotic flagella, which drag their owner through the fluid medium that they are in, flagella propel their owner forward. So as the whip-like flagella waves back and forth, then this pushes the cell in the opposite direction. So it whips back and forth this way, and the cell travels in this direction. Whereas with the eukaryotic flagellum, if this were the cell here, as this whips along, then, or rotates, I should say, then it would pull the cell through the fluid. The structure of the flagella is much more complex than it is in prokaryotic cells. They have this, what is referred to as a 9+2 arrangement, where the microtubules that support the flagellum are built of 9 pairs of microtubules that are in a circle. So if we can see the pairs like this, 2, 3, 4, 5, 6, 7, 8, and 9. So there are the 9 pairs, and they're all around a central pair. So that's the 9+2 arrangement. And so they are, you can kind of make them out here. So we're looking at a cross section. The microtubules would be going on in this direction. And as they slide past each other, as these microtubules slide past each other, then the flagellum whips back and forth. And in general, among eukaryotes, if there is a flagella on it, then there's only one. Cilia have a similar construction as flagella, the same 9+2 arrangement, but rather than being a long whip-like structure, they are very short, and they have a very coordinated kind of movement. They do this stroke recovery kind of movement where the cilia beats forward, push, and then it pulls back and then is upright, beats forward, pulls back, and is upright. So that is, then they use cilia then to propel the cells through the fluid. Generally, they either cover the cell completely, or they're found in these bands of cilia around the cell. And they can have as many as 10,000 or even more cilia on a single cell. So lots and lots of cilia. A third method of locomotion is using something called a pseudopod or pseudopodia, if we're thinking about it in plural form. So these are extensions of the cell membrane. And as the cell membrane sort of pushes forward, then the less dense cytosol, the fluid that is less dense there, kind of moves first into these extensions of the cell membrane. And then the more dense material kind of falls in afterward. So if we imagine that we have this amoeba here, and it's flowing kind of in this direction. So that's terrible. Let's see if I can try this again. So here's the amoeba, and it's moving in this direction. So it will stick out. Just do this dotted, like it'll stick out this extension of the cell membrane. The less dense cytosol immediately rushes in, but then the heavier cytosol kind of falls in afterward, and all the cell contents kind of stream into this pseudopod or false foot that keeps sticking outward. And so lots of complex things can happen with these pseudopodia. They can wrap around, like for instance here we have an amoeba that is about to capture that material. So it sees the material here, it sees it, it senses it, it smells it probably, or tastes it might be a better way to think of that. The pseudopodia reach out around it until it is finally completely enclosed, and now that food item is going to be in a vesicle, so it's just done endocytosis. So amoebae use this as a means of locomotion. There's all about some pseudopods. Okay, so we've seen three means of locomotion in eukaryotic cells: flagella, cilia, and pseudopodia. Last of all, as an external structure, let's talk about cell walls. Cell walls have a very different composition in eukaryotic cells than they do in prokaryotic cells, but they have similar function. They provide rigidity and support for the cell and the cell membrane. We see cell walls in algae and in fungi and in the protist groups. So algae, their cell walls are composed of cellulose, a polysaccharide of glucose, if you recall, from other classes where you might have learned that. So it's this just a bunch of glucose molecules all stuck together, but provides lots of structural support. And so, when we eat lettuce, or when trees are standing tall, they can do all that. That lettuce has fiber in it because of the cellulose. Fungi, their cell walls are composed of a protein called chitin. So it's a protein and polysaccharide. So they use either chitin, or they also can use cellulose as their cell wall material. And then finally, protists, the protozoa, I should say, rather than just protists, but the mobile predatory protists, the protozoa, they have, rather than a cell wall, they have a very flexible kind of supportive covering called a pellicle. So we saw pellicle as a term. I hate the way biologists do this, but we saw pellicle as a term for prokaryotes, that layer that forms right at the water surface. And now we're seeing it again in protists or protozoans having a flexible covering called a pellicle. So where did where did eukaryotic cells come from? Well, the theory that explains that best, and I want to remind you that again of the way that biologists use the term theory. These are very well supported explanations for processes. And so, I think most biologists accept endosymbiosis as the explanatory theory for how eukaryotes arose. It was first proposed, though, in the very early 1900s by a Russian named Konstantin Merezhkovsky. There we are. But he didn't have a lot of evidence to support this. But compelling evidence was provided by a researcher named Lynn Margulis. In 1967, she published the first paper with lots of compelling evidence to support a hypothesis of endosymbiosis. It received a lot of pushback at the time, but since then, and with the work by Margulis as well as many other researchers, it has become generally acknowledged as the explanatory for it. So first proposed by old Konstantin, and really, really put out there as a viable hypothesis by Lynn Margulis. So prokaryotes arose something like 3.4 billion years ago, but we didn't see eukaryotic cells come around until about 1 billion years ago. And we know this not so much from fossil evidence, but instead from estimates looking at differences in the DNA that we find among eukaryotic cells, being able to kind of track back, we come up with these figures. And they're very round figures, of course. But the idea is that a host cell, the idea of endosymbiosis, is that a host cell, via endocytosis, brings another prokaryote into its cell. But rather than digest it, the two live in a symbiotic relationship. So the endosymbiont continues to live It metabolizes and reproduces inside the protected environment of the host cell. So it gets protection, while the host cell is getting some sort of opportunity from having the endosymbiont as well. Perhaps it made a lot of energy, enough energy to support both the endosymbiont and to provide significant support to the host cell as well. And so a mutual relationship arises where both the host and the endosymbiont are living together and each receiving value from the relationship with the other organism. And over time, and with a lot of time, we're talking again, 1 billion years ago, so 1 billion years worth of evolution, the symbiotic cells have evolved into the organelles that we see in what is now considered a eukaryotic cell. So what are some evidence that supports this? What was it that Margulis and her comrades noticed and have since been able to really turn to as explanatory evidence? Well, the first thing is that the size of the mitochondria and chloroplasts that we see in eukaryotic cells are very similar to the size of prokaryotic cells. And mitochondria and chloroplasts both have their own DNA, and it's circular, very similar to prokaryotic DNA, and very dissimilar to eukaryotic DNA. The ribosomes that are found inside mitochondria and chloroplasts have a much similar construction to prokaryotic ribosomes than they do to eukaryotic ribosomes. So ribosomes have the same weight. The book talks a little bit about how you can measure the weight of the subunits of ribosomes, and we see weight ratios that are much more similar to the prokaryotic cells than to eukaryotic cells, with the ribosomes that are inside of mitochondria and chloroplasts. Now, the mitochondria that are outside the chloroplasts and mitochondria but inside the cell, they are more like eukaryotic ribosomes. But the ones that are inside these organelles, they're the ones that look and have qualities very similar to prokaryotes. Also, when we look at protein synthesis inside the organelles, we see that the processes, the biochemical processes of protein synthesis are much more similar to prokaryotic biosynthetic processes than to eukaryotic ones. Again, suggesting that the interior of the mitochondria and chloroplasts, came from a prokaryote. And then when we use antibiotics against eukaryotic cells, and these are antibiotics that stop protein synthesis in bacteria, we see that they also halt protein synthesis not in the whole eukaryotic cell, but inside the chloroplasts and the mitochondria. So again, that's part of why we know these biochemical processes are similar. More evidence. Mitochondria and chloroplasts, when they reproduce, they do it by binary fission, and they do it on a cycle that is independent of the eukaryotic cell cycle. When we look at the membranes of mitochondria and chloroplasts, they are constructed in a way that is much more similar to a gram-negative bacteria's membrane than it is to the eukaryotic cell's membrane in general. So there are enough structural differences between the membrane of the mitochondria and chloroplasts to see that they are different from the eukaryotic cell's membrane and much more similar to gram-negative bacteria. Chloroplast structure is super similar to prokaryotic photosynthetic cyanobacteria. The blue-green algae are this group, which is just one of the groups of archaebacteria. The cyanobacteria are. And then last piece of evidence is that the nucleotides in the DNA found in mitochondria are a very close match to the DNA of a bacteria called Rickettsia provazekii. So Rickettsia provazekii, which is a bacterial species. So again, that tells us, or leads us to believe, that ancestors of Rickettsia provozekii and ancestors of mitochondria were the same, so that they both shared a common ancestor, these mitochondrial to B cells and Rickettsia P. All right, so great, there's a lot of evidence. Can we see any of these kinds of endosymbiotic relationships in the real world today? Well, sure, we do. In the guts of termites, protists there don't have any mitochondria, but instead the protists that are there doing the digestion of the cellulose that termites are known for being able to do. They eat wood, but they can't digest the wood. They rely on protists in their guts to digest it for them. But when you look at those protists, they don't have any mitochondria in them. But they do have bacterial endosymbionts that produce ATP in the same way that mitochondria do. So there's one piece of endosymbiosis can really happen. There's a genus of giant amoebae called Pellicle, and it's found in freshwater ponds, in the muds there in freshwater ponds, and they too don't have any mitochondria, but they have endosymbiotic bacteria that live inside them and produce ATP for them. As well, the protists that are found in termite guts, the ones that have the mitochondria-like endosymbionts, they also have attached to them something on the order of 500,000 spirochete bacteria. And those spirochetes, they are acting like spirally flagella that give the protist the ability to move around. And then finally, the last endosymbiosis that we see is with the prokaryotes that metabolize hydrogen sulfide in deep sea vents. So there are hot water vents in the deep, deep ocean, and a lot of hydrogen sulfide, stinky, smelly stuff, comes out of these hot water vents in the ocean floor, and the bacteria, there are bacteria, let's say that live endosymbiotically with sea tube worms. Tube worms are worms that build these fairly elaborate tubes, and then they can stick their heads out or some parts out one end of the tube and use them to filter feed. But with these tube worms, they have an endosymbiotic prokaryote that lives in their tissues and uses the hydrogen sulfide as an energy source through a process called Chemo Synthesis. So sort of like we talk about photosynthesis, well, chemosynthesis is getting energy directly from chemicals. So 4 examples for you there of endosymbiotic organisms found today around us. And Margulis would suggest to you that they are in the process of evolving into organelles. And here we see a picture, I forget what this picture was now. Oh, dadgummit. You'll have to ask me in class what this is, and I'll try to remember then to be able to explain it. But trying to think. This is the. These are the ATP-producing bacterial endosymbionts that are found in the termites. I think that's what it is. Okay, so we know that, or what we suspect then, is that mitochondria, chloroplasts, and nuclei all came from an endosymbiotic origin, which is interesting to think about the nucleus and the DNA that's in the nucleus being an endosymbiont, coming in, taking over the cell processes and eventually owning the cell and forcing the cell to make its proteins. There's possibility then that these 3 sets of organelles, Golgi apparatus, so the microtubules and microfilaments, And cilia and flagella all also had an endosymbiotic origin. And we already talked about how those protists that are in the guts of termites have all these spirochetes that are acting like little cilia on their surfaces and helping to move the protists around. Okay, so that's everything you ever wanted to know about eukaryotic cells, but might have been afraid to ask. You know how to reach me if you need me.